Rudkin from the University of Aberdeen and Microbiologics Limited, and I'm here to present my poster today, which is on patient-derived monoclonal antibody therapy to treat invasive candida infection. So there's around 1.5 million cases of invasive candida infection globally every year, and around 480,000 directly attributable deaths. And one of the reasons for this is a real lack in treatment options and, of course, rising drug resistance. So there's a real need to develop novel, new mode of action therapies. So we employed a novel single B-cell technology to directly clone the antibody variable heavy and light chain domain genes from patients who had recovered from candida infections. And the result of this was a panel of 18 fully unique human IgG1 monoclonal antibodies. And from this panel and characterization, we've identified antibody 119 as the lead. So to give a brief overview of the technology, we isolated class switch memory B cells from patients who had recovered from infection and screened these against defined protein antigens and candida whole cell preparations, and then directly cloned the variable heavy and light chain domain genes from single B cells, expressed these recombinantly. And as I said, the result of this was a panel of 18 fully human monoclonal antibodies. So antibody 119, our lead antibody, binds to a core N-mannan target in the candida cell wall. And we've shown this here through immunofluorescent imaging and immunogold localization and TEM images. So we know that antibody 119 binds to candida M-mannan. We wanted to make sure that we didn't see any cross-reactivity to other polysaccharides. So we first of all screened a panel of other fungal, bacterial and plant polysaccharides and didn't see any binding to these. And then we also looked at a panel of mammalian N-glycans and didn't see any cross-reactivity to these. So this is really important in terms of de-risking antibody 119 as we go forward. Antibody 119 has broad candida species coverage. So by that I mean we've looked at testing its binding to a panel of candida species, including Candida auris, Candida parapsilosis, Lusitaniae, glabrata, and we see good binding to all of these clinically relevant isolates. And we've shown this through immunofluorescent imaging and then also through flow cytometry as well. In terms of the mechanism of action, one of the ways antibody 119 exerts its activity is through recruiting immune cells to clear fungal cells through phagocytosis. So we've shown this through live cell imaging, and we've done this for Candida albicans, Candida auris, Candida glabrata, and also looked at drug-resistant and drug-sensitive isolates. And we have demonstrated that when antibody 119 is present and bound to the candida, we see a significant early clearance of the fungal cells from the assay. So following on from the opsonization of fungal cells for phagocytosis, this translates into protection in vivo models of systemic infection. So first of all, we looked at the Galeria melanella model, and we've shown here, looking at drug-resistant Candida albicans, Candida auris, and Candida glabrata isolates, that we can significantly improve survival in this infection model. And that's shown here on the graph by the blue lines. We've also looked at mouse models of infection. So we've done a prophylactic study where we administered antibody 4 hours prior to infection with Candida albicans. And we demonstrated a significant reduction in kidney fungal burden versus an IgG1 control antibody in this model. So just to summarize our work, we generated a panel of fully human IgG1 monoclonal antibodies, and from these we identified antibody 119 as the lead antibody to take forward. We've shown that this antibody binds to core N-mannan in the candida cell wall and primarily works through opsonizing fungal cells for phagocytosis. And this translates into protection in vivo models of systemic infection. So we're currently looking at additional mechanisms of action and also antibody sequence optimization as well. So hopefully we're going to be publishing some of that work later this year. So finally, I'd just like to acknowledge all the other team members at the University of Aberdeen, Microbiologics Limited, and our collaborators as well as the funders, particularly the Wellcome Trust, who are currently funding us at this latest stage of the programme.